Hey students, I'm your Nandini Ma'am. Today, I'm going to read Chapter 2, Biological Classification of Class 11, Biology, NCERT. So let's get started. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdoms was developed that included all plants and animals respectively. This system was used till very recently. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plants and animals was easily done and was easy to understand. In spite, a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was found inadequate. A need was also felt for including, besides gross morphology, other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification systems for the living organisms have hence undergone several changes over time. Though plant and animal kingdoms have been a constant under all different systems, the understanding of what groups or organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over time. R. H. Whitaker proposed a five-kingdom classification. The kingdoms defined by him were Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, thallus organization, mode of nutrition, reproduction, and phylogenetic relationships. Table 2.1 gives a comparative account of different characteristics of the five kingdoms. Let us look at this five kingdom classification to understand the issues and considerations that influence the classification system. Earlier, classification systems included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and the angiosperms under plants. The character that unified this whole kingdom was that all organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae with other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones. Say, for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group, the fungi, and the autotrophic green plants, though they also showed a characteristic difference in their walls composition. The fungi had chitin in their walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, Kingdom Fungi. All prokaryotic organisms were grouped together under Kingdom Monera, and the unicellular eukaryotic organisms were placed in Kingdom Protista. Kingdom Protista has brought together Chlamydomonas, cholera, with paramecium and amoeba, which lack it. It has put together organisms which in earlier classifications were placed in different organisms or kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities, but is also phylogenetic. That is, it is based on evolutionary relationships. In this chapter, we will study characteristics of kingdoms Monera, Protista, and fungi of the Whitaker system of classification. The kingdoms Plantae and Animalia, commonly referred to as plant and animal kingdoms respectively, will be dealt with separately in chapters 3 and 4. Kingdom Monera. Bacteria are the sole members of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans where very few other life forms can exist. Many of them live in or on other organisms as parasites. Bacteria are grouped under four categories based on their shape: the spherical coccus, the rod-shaped bacillus, the comma-shaped vibrio, and the spiral spirillum. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs. That is, they do not synthesize their own food but depend on other organisms or on dead organic matter for food. Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas, halophiles, hot springs, thermoacidophiles, and marshy areas, methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the guts of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane from the dung of these animals. Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have chlorophyll A similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial, or filamentous, marine, or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts, for example, nostoc and anabina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are the most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume, roots, etc. Some are pathogens causing damage to human beings, crops, farm animals, and pets. Cholera, typhoid, tetanus, citrus canker are some well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Sometimes, under unfavorable conditions, they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. The mycoplasmas are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Kingdom Protista. All single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protistium to one biologist may be a plant to another. In this book, we include gyrosphites, dinoflagellates, euglenoids, slime molds, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the others dealing with plants, animals, and fungi. Being eukaryotes, the protistian cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. Gyrosphites. This group includes diatoms and golden algae. They are found in fresh water as well as in marine environment. They are microscopic and float passively in water currents. Most of them are photosynthetic. In diatoms, the cell walls form two thin, overlapping shells which fit together as in a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producers in the ocean. Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on the main pigments present in their cells. The cell wall has stiff cellulose plates on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the wall plates. Very often, red dinoflagellates, for example, Goniacolex, undergo such rapid multiplication that they make the sea appear red. Toxins released by such large numbers may even kill other marine animals such as fishes. Nucleanoids. Majority of them are freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigments of euglenoids are identical to those present in higher plants, for example, euglena. Slime molds. Slime molds are saprotrophic protists. The body moves along decaying twigs and leaves, engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms fruiting bodies bearing spores at their tips. The spores possess true walls, they are extremely resistant and survive for many years, even under adverse conditions. The spores are dispersed by air currents. Protozoans. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in fresh water, seawater, or moist soil. They move and capture their prey by putting out pseudopodia as an amoeba. Marine forms have silica shells on their surface. Some of them, such as entamoeba, are parasites. Flagellated protozoans. The members of this group are either free-living or parasitic. They have flagella. The parasitic forms cause diseases such as sleeping sickness, example trypanosoma. Ciliated protozoans. These are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity that opens to the outside of the cell wall. The coordinated movement of rows of cilia causes the water laden with food to be steered into the gullet. Example, paramecium. Sporozoans. This includes diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, which causes malaria, which has a staggering effect on human population. Kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. When your bread develops a mold or your orange rots, it is because of fungi. The common mushroom you eat and the toadstools are also fungi. White spots seen on mustard leaves are due to a parasitic fungus. Some unicellular fungus, for example yeast, are used to make bread and beer. Other fungi cause diseases in plants and animals. Wheat rust causing leukemia is an important example. Some are the sources of antibiotics, for example penicillin. Fungi are cosmopolitan and occur in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infections. With the exception of yeast, which are unicellular, fungi are filamentous. Their bodies consist of long, slender, thread-like structures called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called coniocytic hyphae. Others have septae or cross walls in their hyphae. The cell walls of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophs and absorb soluble organic matter from dead substrates, and hence are called as saprophytes. Those that depend on living plants and animals are called parasites. They can also live as symbionts in association with algae as lichens and with roots of higher plants as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spores called conidia or sporangiospores or zoospores. And sexual reproduction is by oospores, ascospores, and basidiospores. Various spores are produced in distinct structures called fruiting bodies. The sexual cycle involves the following three steps. Fusion of protoplasms between two motile or non-motile gametes called plasmogamy. Fusion of two nuclei called karyogamy. Meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, two haploid hyphae of compatible mating types come together and fuse. In some fungi, the fusion of two haploid cells immediately results in diploid cells. However, in other fungi like Ascomycetes and Basidiomycetes, an intervening dikaryotic stage occurs. Such a condition is called a dikaryon and the phase is called dikaryophase of fungus. Later, the parental nuclei fuse and the cells become diploid. The fungi form fruiting bodies in which reproduction division occurs, leading to formation of haploid spores. The morphology of the mycelium, mode of spore formation, and fruiting bodies form the basis for the division of the kingdom into various classes. Phycomycetes. Members of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places or as obligate parasites on plants. The mycelium is aseptate and coniocytic. Asexual reproduction takes place by zoospores or by aplanospores. These spores are endogenously produced in sporangium. Zygospores are formed by fusion of two gametes. These gametes are similar in morphology or dissimilar. Some common examples are mucor, rhizopus, and elbobo. Ascomycetes. Commonly known as sac fungi, the Ascomycetes are unicellular, for example, yeast, or multicellular, like penicillin. They are saprophytic, decomposers, parasitic, or coprophilous, which are growing on dung. Mycelium is branched and septate. The asexual spores are conidia produced exogenously on the special mycelium called conidiophores. Conidia are on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like asci. These asci are arranged in different types of fruiting bodies called ascocarps. Some examples are aspergillus, claviceps, or neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and bacillus are edible and are considered delicacies. Basidiomycetes. Commonly known forms of Basidiomycetes are mushrooms, bracket fungi, or puffballs. They grow in soil, on logs and tree stumps, and in living plant bodies as parasites, for example, rust and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis take place in the basidium, producing four basidospores. The basidiospores are exogenously produced on the basidium. The basidia are arranged in fruiting bodies called basidiocarps. Some common members are agaricus, Ustiligo, and Puccinia. Deuteromycetes. Commonly known as imperfect fungi because not only the asexual or vegetative phases of these fungi are known, when the sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage have been given one name and the sexual stage another. Later, when the linkages were established, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect stages of members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual spores known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, called Telecotrichum, and Trichoderma. Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll-containing organisms commonly called plants. A few members are partially heterotrophic, such as the insectivorous plants or parasites. Bladderwort and Venus flytrap are examples of insectivorous plants, and Cascata is a parasite. The plant cells have an eukaryotic structure with prominent chloroplast and cell wall mainly made of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plantae includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Life cycle of plants has two distinct phases, the diploid sporophytic and the haploid gametophytic that alternate with each other. The lengths of the haploid and diploid phases and whether these phases are free-living or dependent on others vary among different groups in plants. This phenomena is called alternation of generation. You will study further details of this kingdom in chapter 3. Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cells lack cell walls. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserves as glycogen or fat. Their mode of nutrition is holozoic by ingestion of food. They follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. The salient features of various phyla are described in chapter 4. Viruses, viroids, and lipids. In the five kingdom classification of Whittaker, there is no mention of some acellular organisms like viruses, viroids, and lipids. These are briefly introduced here. All of us have suffered the ill effects of common cold or flu, know what effects viruses can have on us, even if we do not associate it with our condition. Viruses do not find a place in classification since they are not truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Once Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call viruses living or non-living? The name virus, that means venom or poisonous fluid, was given by Pasteur D.J. Ivanescu, recognized certain microbes as casual organisms of the mosaic disease of tobacco. These were found to be smaller than bacteria because they pass through bacteria-proof filters. N.W. Bain-Jernick demonstrated that the extract of the infected plants of tobacco could cause infection in healthy plants and called the fluid as Contagium vivum fluidum. W. M. Stanley showed that viruses could be crystallized and crystals consist largely of proteins. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to proteins, viruses also contain genetic material that could be either RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages are usually double-stranded DNA viruses. The protein coat called capsid, made of small subunits called capsicomeres, protects the nucleic acid. These capsomeres are arranged in helical or polyhedral geometric forms. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by a virus. In plants, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, drafting, and stunted growth. Viroids. In 1971, T.O. Diner discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein coat that is found in viruses, hence the name viroid. The RNA of the viroid was of low molecular weight. Lichens. Lichens are symbiotic associations, that is, mutually useful associations between algae and fungi. The algal component is known as phycobuoyant and the fungal component as mycobuoyant, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrients and water for its partner. So, close is their association that if one saw a lichen in nature, one would never imagine that it had two different organisms within them. Lichens are very good pollution indicators. They do not grow in polluted areas. Summary. Biological classification of plants and animals was first proposed by Aristotle on the basis of simple morphological characters. Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Whitaker proposed an elaborate five-kingdom classification, Monera, Protista, Fungi, Plantae, and Animalia. The main criteria of the five-kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationships. In the five-kingdom classification, bacteria are included in kingdom Monera. Bacteria are cosmopolitan in distribution. These organisms show the most extensive metabolic diversity. Bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista includes all single-cell eukaryotes such as chrysophytes, dinoflagellates, nucleanoids, slime molds, and protozoans. Protists have defined nucleus and other membrane-bound organelles. They reproduce both asexually and sexually. Members of kingdom Fungi show a great diversity in structures and habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual reproduction. Phycomycetes, Ascomycetes, Basidiomycetes, and Eutromycetes are the four classes under this kingdom. The Plantae includes all eukaryotic chlorophyll-containing organisms. Algae, bryophytes, rhidophytes, gymnosperms, and angiosperms are included in this group. The life cycle of plants exhibit alternation of generations, gametophytic and sporophytic generations. The heterotrophic eukaryotic multicellular organisms lacking a cell wall are included in the kingdom Animalia. The mode of nutrition of these organisms is holozoic. They reproduce mostly by the sexual mode. Some acellular organisms like viruses and viroids are, as well as the lichens, are not included in the five kingdom system of classification. Thank you all.